So welcome back friends, welcome back in another video tutorial from microbiology with Sharnik. So in this video tutorial we will going to discuss about our last topic of application in recombinant DNA technology, that is vaccine formation with the help of recombinant DNA technology. Okay, so as we have get a huge success in every kind of work that we have done with the help of recombinant DNA technology, so now it become necessary to also use recombinant DNA technology in the field of vaccine formation, because you know what, earlier times we are using just the exact organism as a vaccine and we are injecting it inside our body after doing some modification in it. This modification is nothing but we are making it inactive, right? So inside the body our immune system will recognize those kind of inactive organisms or inactive pathogens or inactive virus, whatever you can say, and our immune system will construct its own defense system for particularly for that type of organisms, right? So as a result, whenever our body further face that same pathogen or same viruses, it gets activated fastly, right? That is the basic funda of vaccine formation, right? But in earlier times, before the entry of recombinant DNA technology in that field, there was a problem, because you know what, we are using or we are inserting simply the entire pathogen, right? So it causes some type of problem because we are also inserting some unnecessary things, right? And they may have some negative effect in our body. So it becomes necessary to only insert the part of the virus or let's say bacteria that is responsible for the immune response generation inside our body. Okay, so as scientists find out that they have restriction in the nucleus and various type of tools like this that have the ability to only isolate that part of the body that is responsible for immune response generation in our body, so it's become necessary to use recombinant DNA technology in that field, right? So in this video we will going to discuss about viral vaccine formation, because recombinant DNA technology is mostly used for that purpose only. So here we will going to discuss about hepatitis B viral vaccine formation. Okay, so under such circumstances, just like our previous videos, we will going to first discuss about what is the problem that is going on, and we will try to find out what can be a preferable solution for that problem. Okay, so hepatitis B viral vaccine formation, that means here we are constructing the vaccine against hepatitis B virus. Okay, so now it is important to know about what hepatitis B causes inside our body. So hepatitis B causes an inflammatory contagious liver infection. Okay, so that is the idea. So now it becomes necessary to prevent it because it causes various kinds of problems, you know. For example, jaundice, for example, lymphoadenopathy, that is not lymphoadenopathy, but it is necessary that it causes hepatomegaly. Hepatomegaly means, as we all know, that in our liver cells is known as hepatocyte. So due to the entry of the hepatitis B virus, there is a chance of increasing the size of the our liver, that is known as hepatomegaly, right? So now it also causes liver cancer, right, under some chronic infective situation. So it is necessary to construct viral vaccine against that particular type of virus, right? So now first thing is that we have to know about something about the structure of the hepatitis B virus. Okay, so this structure will help us to determine what part of the body we should choose for our immune response generation. Okay, so let's see about the structure first. Okay, so this is the structure of the hepatitis B virus. So as you can see that it is DNA virus. Okay, but there is a special type of speciality associated with this, that is that the DNA molecule is partially double stranded instead of fully double stranded. Okay, and it also contains a DNA polymerase by its own, because remember it, as it contains a unusual type of DNA that our normal cell do not contain, so it is necessary for it to contain its own enzyme to perform its replication. Okay, so now the important thing is that we have to know that it contains three type of antigen. One is hepatitis B envelope antigen, hepatitis B core antigen, and hepatitis B surface antigen. Okay, so now this, as this surface antigen is the outside of the hepatitis B, we will going to target hepatitis B surface antigen to construct the hepatitis B viral vaccine. Okay, so let's see how hepatitis B viral, we are using hepatitis B surface antigen to construct hepatitis B viral vaccine. Okay, so this is the hepatitis B viral vaccine, as you hepatitis B virus, as you can see. So I have already conducted a video lecture regarding the gene library. Okay, so I want to told you that there is also gene library available for hepatitis B virus. Okay, so the first goal will be to isolate the gene which is responsible for the production of the hepatitis B surface antigen. Okay, so we have isolated it with the help of colony hybridization techniques that we have discussed. So if you want to know about what is colony hybridization, what is plaque hybridization, what is colony PCR, that help us to find out our gene of interest from a library. So I would like to recommend you to visit my playlist that I have created on the basis that is totally focused on the construction of genomic DNA library and cDNA library. So if you go through those videos, then I hope you can understand about everything regarding gene DNA library and how you can isolate a particular gene from gene DNA library. Okay, so now it is clear that we are wanting a protein, right? So the vector should be a expression vector. Okay, so according to the normal property of vector, it should have a promoter for transcription, a transcription terminator site, a yeast origin of replication, a LEU2 that is a specific part of our yeast vectors, and a tetracycline resistant part, and a bacterial origin of replication. That means not only we are using a expression vector, but it is as well as a shuttle vector too, right? So remember, so first of all we will insert them here with the help of DNA ligase. Okay, so now after that, once we have our vector ready, we will transform them inside the yeast. Okay, so here we will prefer yeast because we are constructing a protein of virus. Okay, so then we will grow them in fermenter in the absence of leucine, because you know this picture is that I have taken to explain the basic concept of the hepatitis B viral antigen viral vaccine formation is specifically focused on the hepatitis B viral vaccine formation on the industrial scale level. That is why they have used as a fermenter. Okay, so in the absence of leucine, we will allow those yeast to grow in the fermenter. Okay, then after that we will isolate hepatitis B surface particles from the whole cell extract. Then we will formulate them into a vaccine, and the formulation is also necessary, important. I will going to discuss about it. So we are using a yeast vector, right, that should contain a promoter, a transcription terminator, a yeast origin of replication, bacterial origin of replication, a bacterial selection marker, that is tetracycline resistance gene, and another one is LEU2 gene. Okay, then we will insert that hepatitis B surface antigen there and then transform them into yeast. Okay, then we will allow those yeast to grow in fermenter in the absence of leucine. Then we will isolate hepatitis B surface. Okay, so there was a problem, so no problem. So now So we are growing them in fermented in the absence of leucine. Then we will isolate them in hepatitis B surface antigen. Then we will formulate into vaccine. So now we will learn about how hepatitis B vaccine is formulated. Okay, so let's go back into the PDF. So the solution that we have obtained, it is a sterile solution of immunoglobulin containing antibodies that is specifically against hepatitis B surface antigen. Okay, and it is possible to done because the hepatitis B surface antigen that we have obtained with the help of those fermenters that we have constructed. Now it is necessary to inject those kind of antigen in another kind of animals, for example, let's say mice. So the mice immune system will generate the antibody for it. Okay, that's how we can supply the ready-made antibody for our immune system. Okay, but in some cases we are also using the direct antigen. Okay, that means the solution, the sterile solution, in some cases can be only immunoglobulin containing only a solution containing hepatitis B surface antigen, and in some cases it will be a sterile solution of immunoglobulin containing antibody against hepatitis B surface antigen. So we will keep them in pH 6.2 and we will use various kind of solution for the formulation. They are 0.075 molar NaCl, 0.15 molar glycine, 0.01% polysorbate 80, and we will use those kind of solution by intramuscular route as a vaccine. Okay, so what kind of solution we are using for formulation of the vaccine is necessary to understand. That are 0.075 molar NaCl, 0.15 molar glycine, 0.01% polysorbate 80. Okay, and we are using intramuscular route as a vaccine for hepatitis B. Okay, so now we have discussed about the preparation. You can use yeast, you can use another mammalian cell line. For example, you can use Chinese hamster ovary cells and other kind of suitable cell lines. Okay, so we will construct a sterile solution that may contain only the antigen, or that may also contain the immunoglobulin containing antibody against hepatitis B surface antigen, and the pH will be at 6.2. Okay, and then we will formulate the solution with the help of 0.075 molar NaCl, 0.15 molar glycine and 0.01% polysorbate 80. Okay, so we will use them via intramuscular route. So we can use only Saccharomyces cerevisiae or another kind of mammalian cells, for example Chinese hamster ovary cells or other suitable cell lines. So that was the concept of the hepatitis B viral vaccines. So remember here very carefully that we are using hepatitis B surface antigen for the construction of the virus, construction of the vaccine. Okay, so we can either inject the antigen inside the body directly so that our immune system can make appropriate antibody against that antigen, or we can also provide direct ready-made antibodies also, right? And we are using various kind of Chinese hamster ovary cells or other kind of mammalian cell lines, or let's say Saccharomyces cerevisiae cell for the construction of the hepatitis B virus vaccine. Okay, so that was the concept of the hepatitis B virus vaccine formation. So I hope this video will be helpful to you, and thank you for listening to this class. Bye bye.